Thank you very much. Immusoft is developing a transformational cell therapy platform that uses a patient's B cells as biofactories for sustained protein delivery. More specifically, we're working with plasma cells, which are a subset of B cells. And I like to think of plasma cells as kind of like the immune system's dedicated biofactories. So they're producing and secreting thousands of antibodies per second. And it's that biofactory ability of the plasma cells that we harness by putting a therapeutic gene into those cells and enabling them to produce our therapeutic protein then at mass quantity. The idea being we will infuse those into the patients. The plasma cells will engraft into the bone marrows where they remain for many years, producing our therapeutic proteins over the life of that plasma cell. So, when I think about systemic gene or protein delivery, there are really two kind of high-tech modalities to do that. One is the AAV-based gene therapy, which involves systemic injection of a virus, and the other is stem cell mediated gene delivery. And we really are bringing a third modality to the table with our plasma cell platform. With these plasma cells, they're virus-free, so we avoid the immunogenicity that's associated with virus delivered systemically. And we don't require the myeloablation that's associated with the stem cell procedures. So we've got substantial advantages really over both. And we've used our approach to now deliver a multitude of different types of proteins, those being enzymes, antibodies, signaling proteins, and I'm sure you can appreciate that under each of those modalities there are numerous potential indications that we could pursue. So it's truly a platform that we're excited to be advancing. Our lead indication is MPS I. MPS I is a lysosomal storage disease. We're advancing rapidly towards the clinic, looking to enroll our first patient this quarter. We've got great preclinical data, and the preclinical data, along with knowledge of recombinant enzymes that are already on the market in this space, really give us an indication that we should see proof of concept pretty early on in a clinical study, so within weeks of dosing. Moving to the management team, it's a group that I'm just really proud to be working with. They're dedicated, they're serious about having a positive impact on human health, and they show up. They bring it every day. Great group. And so, as far as the two other modalities that I mentioned, stem cell mediated, virus mediated. Again, there's no need for myeloablation with our procedure. Plasma cells naturally engraft in the bone marrow. Systemic virus delivery, we mitigate we have no expectation of the immunogenicity that's associated with virus delivery. And so what that means is, at the outset, let me take a step back. In the case of virus delivery in particular, we all have, usually there are various levels of antibodies in each of us to the virus that's being injected. And so what that means is we're trying to get a one-size-fits-all dose. If it's wrong the first time, there's really no opportunity to go back and redose those patients. But with our approach, we can titrate up, we can give a dose, monitor, give a new dose if need be, and then in the out years, if the efficacy or the protein expression begins to wane, there's an opportunity for us to deliver again. And moreover, we're delivering a defined cell population. So plasma cells are CD138 positive. There's an antibody on the market that is designed to selectively remove CD138 positive cells. So in the case that we need to reduce or eliminate the plasma cells, we've got the opportunity to do that. And there's really, those are two elements of kind of fine-tuning, I like to think of it, that other modalities of gene therapy really cannot claim. And so the other current modality for systemic delivery of proteins is enzyme replacement therapy. And we think we've got pretty substantial advantages over enzyme replacement therapy as well. not the least of which is these enzymes are delivered by infusion. It takes 4 roughly hours per week that this has to be done over, and that involves usually the patient who's a young child plus a caregiver that has to bring that child to the infusion clinic. So by having sustained delivery of the enzyme, we're really giving that time back to these families. And then in terms of just the efficacy. In enzyme replacement therapies, they're delivered on a weekly basis. The patient goes in, the glycosaminoglycans, in the case of MPS I, they get knocked down. A week later, they're back up again, and you knock them down, and you go through that cycle. You can't keep them down, but with our approach, we have sustained delivery of the enzyme, sustained reduction of glycosaminoglycans, which we think will ultimately yield better outcomes. And so the way the approach works is we start at the apheresis site. The patient is apheresed. We then ship that product to our processing facility where the B cells are separated. They're then transposed with our Sleeping Beauty transposon. So again, this is a virus-free approach. We expand the B cells. We differentiate them into the plasma cells and then inject them into the patient where we expect they'll engraft and produce a protein for many years. I would point out that our culture system is 7 days. And by contrast, CAR Ts are usually 2-3 weeks. And the implications are that that culture system is really one of the bigger cost drivers in autologous approaches. And so by having ours down to 7 days, I think that gives us a nice cost point. I mentioned that we've delivered multiple different protein types, enzymes, antibodies, and so forth. We're also now delivering multiple proteins under a single construct, which we're really excited by. And that's enabled by the plasmid, the transposon that we're using, which has a much higher cargo payload than virus delivery of a gene. And so just if we, as I mentioned, all these different protein types, they've got a multitude of different indications that we could pursue. If we wanted to just kind of zero in on the enzyme replacement market, this is very compelling for us insofar as these are all recombinant proteins. They're indications that are currently being treated with recombinant proteins by infusion. And so we're starting with MPS I. We know that we have a pretty good idea that our enzyme, iduronidase, is safe and efficacious. And so with some initial proof of concept in MPS I, that will give us the impetus to start moving through and I'll say tackling more of these enzyme replacement approaches. And so a little bit more about our Sleeping Beauty transposon system. It's really got substantial advantages over virus delivery. The primary, I would say, is the avoiding the immunogenicity elements of viruses. Again, the payload is larger, the cost is much lower, lead times to get the product are much shorter. And just simply in terms of complexity, the manufacture of plasmid is much more reproducible, much more stable than virus. So many advantages, and I would point out that also the Sleeping Beauty transposon is being used in a clinical setting with CAR Ts at MD Anderson. So a great safety profile so far as well. So looking at our clinical plans, our pipeline, we are, as mentioned, looking to enroll our first patient in Hurler syndrome this quarter, which we're very excited about. From there, we'll move to muscular dystrophy and MPS II. And I would point out, in the case of muscular dystrophy, we're actually using a protein known as follistatin. And so we're not necessarily looking to correct that dystrophin gene, as most approaches are. But I look at follistatin as kind of a general muscle building protein. So it's something that when we work out, we release that from our liver. It's secreted into the bloodstream and is involved in muscle building. And so we're enabling plasma cells to release follistatin into the bloodstream with the expectation of increased strength and muscle mass. And I would say Hunter syndrome, it's got many similarities and dovetails very nicely with our MPS I program. And so a little bit about MPS I. It's a lysosomal storage disease. It's characterized by the loss or deficiency of the iduronidase enzyme. Iduronidase is responsible for the breakdown of glycosaminoglycans, or GAGs. And when those GAGs build up in the tissues, they cause organ dysfunction, organ damage, and if not properly treated, lead to early death. So there are currently two modalities on the market to treat MPS I. There's a recombinant enzyme, Aldurazyme, and there's stem cell transplants. Aldurazyme, it does cost about $250,000 to $400,000 a year per patient. It does achieve sales in the realm of about $250 million a year, so commercially impactful. Stem cell transplant, it's still, there's a high degree of mortality, so it's usually reserved for the more serious Hurler patients. But I would say that in both cases, neither stem cell transplants nor enzyme replacement therapies are by any means completely curative. So these patients still have plenty of room for upside, and we think that our approach will certainly be an improvement. And this is really a snippet of some of the preclinical data that we've developed in iduronidase for reduction of glycosaminoglycans. The graph, in this case, we compared two different routes of administration, IP and IV. And just to be clear, for our human clinical study, we'll go IV, but we were kind of just interested in doing some compare and contrast, and we were encouraged by the fact that we saw reduction in GAGs in both settings. And so what we have is, on the very left are non-treated animals. These vertical bars each represent a different tissue type, and so we see that middle group that is IV treated, we saw a substantial reduction in GAGs, and then the far right group is IP treated and also a substantial reduction in GAGs. Our MPS I clinical development timeline. 2018 was a good year to get some of the early blocking and tackling done. We achieved Orphan drug designation. We also got rare pediatric disease designation. We started our IND-enabling tox and GMP manufacturing, which we completed earlier this year, and we've now got our IND on file and are looking forward to getting into the clinic. This star is really to call out that first potential sign of biological activity, which I would define as a measurable increase in the enzyme in the blood and/or reduction in glycosaminoglycans. So that initial sign of biological activity is going to be really a tremendous value inflection point for this program, but also not just for this program, but for our platform more broadly speaking. And so I mentioned follistatin as a potential treatment in muscular dystrophy. It's also not genotype specific. And so what that means is that there's also potential for application in other muscle degenerative diseases that might be neuromuscular diseases, or even sarcopenia, which is age-related muscle wasting. So this is a small test, but very encouraging. We saw increases in strength across all three strength tests, and we also saw increases in the muscle mass, or the body mass, which we attribute to increases in muscle mass. I mentioned, just a phenomenal team to be working with. Great experience in cell and gene therapy, companies like Amgen, MedImmune, 5', J&J, Retrosense, Dendreon. I'm the CEO. I'm a serial entrepreneur. I started Retrosense Therapeutics. We took that into the clinic and sold it to Allergan in 2016. I was also deeply involved in the launch of Compendia Bioscience, which we ultimately sold to Life Technologies in 2012. In both instances, it was a great outcome for both the investors and the founders. So this is a space that we really have a dominant intellectual property position in. I would define the space as B cells, the manufacture and or use of B cells as biofactories for sustained protein delivery. We also have freedom to operate. Our IP, it started with some IP out of Caltech from David Baltimore around the culture system. We've been building on that. We've in-licensed some patents from University of Minnesota, Max Delbruck University, and we continue to expand on the IP portfolio with patents that we own in-house, and we are getting continuous notices of allowance and issuances. So very excited about that. So to summarize, we are really the world leader in the space of B cells for biofactories, the only commercial entity in this space. We've got a dominant IP position. We're bringing really a new modality to systemic cell and gene systemic protein or gene delivery. Our modality, it gets around, obviates the issues associated with immunogenicity for viruses, myeloablation with stem cells, and so some clear advantages over both. Our lead asset, we're approaching the clinic with MPS 1. We raised earlier this year $23 million. We closed on anyway in our Series B, and that gets us pretty far into our phase 1 clinical study. But nonetheless, we're certainly interested in talking about partnership opportunities or other strategic opportunities. And so here's my email. Please feel free to contact me if interested. Thanks very much.